it's a great honor for me to be able to participate in all 10 of the Asian perspectives, and in sitting through the meeting so far, I was thinking back and I realized that everything I am going to tell you today is new from 10 years ago. So there has been lots of change, and the change has been positive. And the first thing that I want to point out is that Shishan Yuan is wearing a tie today. That is new. Okay. So I'm talking about the control of PRRS today, particularly emphasizing immunological control. And the question I was asked to address is this something worth doing? Or is immunological control unattainable and cannot be done? A fool is a person who works on something that cannot be done. So that is the question today. And I hope to convince you by the end that no, it is not a fool's errand. We have learned important things about how immunity works in PRRS that shows us that immunity plus other management techniques is successful in controlling PRRS. My university wants me to point out to you that I have a consulting relationship with Boehringer Ingelheim, and the university manages it to avoid conflicts of interest. So the first point in looking at immunity to PRRS virus is to ask, well, what is PRRS virus? Because that's what immunity is directed towards. And we have this impression that it's a tremendously genetically diverse virus, and that any one virus is very different from any other virus. Well, this is a dendrogram of about 7,000 PRRS viruses taken from actual outbreaks on US farms. Major producers sequence viruses so often that over a 15-year period, 5 large producers have generated over 7,000 sequences. And we see a very interesting pattern here. We see evidence of genetic diversity in some areas, but we also see large regions with tens to hundreds of isolates that do not change over a period of here 5 years, here 14 years, here 2-year period. So we see 2 patterns of genetic diversity. One where there's very little, suggesting that viruses emerge that are well adapted, apparently, and can persist unchanged for periods of time, whereas other examples where virus changes rapidly. I will point out that what we call 144 viruses is where NADC30 is located. This is a virulent group of viruses that appeared around 2009, but we no longer see it. These isolates stopped in 2014. What came more recently are what we call 174 viruses. It's possible that this will be the next emerging virus in Asia. Okay. So there are selection pressures that definitely drive PRRS virus evolution, but they are complex and we do not really understand what they are at this time. This ability to evolve and diversify is present throughout the genome of the virus. Here's Sean Wan, a graduate student from Jilin University in my lab, did high throughput sequencing of a field isolate to ask what the genetic diversity was at each base along the genome. And he found that while at any given position you almost always found the same base, about 1 time out of 1,000 you would see a different base. So this background level of genetic diversity is present throughout a population of viruses. Okay. This diversity is always present in populations of PRRS virus. Scientists assume that if you clone a virus, you make a clone. By definition, that word means that everything is the same. But if you and you make PRRS virus clones by genetic engineering and produce it initially in bacteria, but eventually you have to put that nucleic acid into a cell and grow up viruses. When you look at those viruses in passage 1, you see this background of variation across the entire genome. When you passage that group of viruses again, you see the variation. So even at the very beginning, these clones are clouds, a swarm of mutants. And if you look in field isolates, you see the same thing. The scales are different here, but you have this background variation, and you have occasional positions where there's a very high frequency of non-dominant sequences or bases. What we're looking at here is average variation across the genome, but the real question is the entire genomes, are they different or the same? Because here you could have one base in one sequence that's different and the other 14,999 bases are the same. So every time you take a genome and you have a different base, that is called a haplotype. And Sean Wan has done a haplotype analysis of the ORF5 gene and the ORF3 gene in a virulent field isolate, and he sees a population pattern that is the same as has been observed previously in HIV infection in humans. So we see on the x-axis, every dot is a different sequence. And the frequency of that sequence in the population is on the x-axis. So you see a dominant sequence. In ORF5, 45% of all the viruses have a single sequence. The next most abundant haplotype is 9% of the entire population. So you see this distribution where a small number of sequences is most of the population, but then you have thousands of different sequences that are present at very, very low frequencies. This is important because the way PRRS virus outbreaks occur is that virus grows in a pig to a large number, and a very small number of viruses is transmitted to start a new infection in the next pig. That new pig is a new environment. The viruses that go in could be the dominant virus, could be one of these rare viruses that you will not see by standard characterization, so you do not even know it is there. Second, the next pig has a new immunological environment, a new set of conditions, so that the dominant virus in one pig may not survive to be the dominant virus in the next pig. This experiment to ask that question was actually done many years ago at Iowa State by Kunjin Yoon and Jeff Zimmerman. So they cloned a virus. Back then we assumed it was all the same and called it CC-01. They put this virus into pig A on passage 1, and we'll see then they transfer what grows in that pig to a second pig, which is A2, and continued on 7 times. When they put 01 into pig A, waited 60 days, and sequenced 15 clones, they found 01 was still present, but it now was not even dominant. There were other viruses there. When they took this population of viruses and passaged it the second time, the original virus was gone. They could not see it. And the population was dominated by a different sequence, AV01. And by the 7th passage, this virus was still present in a minor population, but they had sequences that had not been seen previously. They were very good scientists. They repeated this 3 times. If we look at the 3rd replicate, half of the viruses in the first passage were the original infectious clone. By the time they got to the 7th passage, they had all of the clones were a single virus type, but that type had not been seen in any of the other pigs that had been examined. So we see this, that this PRRS virus is a swarm of mutants. Okay? When you look in the field, this pattern has been observed in large systems, of which there are several in China today. Dale Polson at Boehringer Ingelheim collaborated with a large system over a period of 7 years. Every time they found a PRRS virus by PCR screening, they sequenced it. The very first virus is shown here in the corner, and this is the percent difference of all of the viruses to the first one. The second time they found this virus, it was a different sequence. Over 7 years, they never saw the original sequence again. They saw this swarm of variation that sometimes stayed the same, sometimes changed, and over time there was this gradual increase in diversity. So the virus does change. That's one pattern. During this 7 years, they found another virus came into the herd. The pattern was slightly different. The same sequence was observed for about 1.5 years, but then again it started to change. And again during this time a third isolate was found, and you see this pattern of variation. You consistently see an increase in variation, but the dynamics of change are very unpredictable. Okay. So we were learning. We did not know 10 years ago that PRRS viruses are swarms of mutants in a pig. A lot of variation. When the virus is transmitted, it's a small number that can be randomly selected from that population, so it's hard to predict what the outcome of transmission will be. It could be the same, which might explain how many outbreaks can occur and the virus seems not to change, or it could jump quickly because of the random nature of what gets transmitted. Okay. One of the conclusions is, as Dr. Yan pointed out, new types of viruses are certain to emerge. Okay? But what will they be? When will they be? Where will they show up? We do not know. Okay. So that's what we are facing and trying to control PRRS virus immunologically. Now I'd like to talk about things we are learning about the ability of pigs to innately mount an antiviral immune response, and then look at the specific anti-PRRS virus response. We reported several years ago that there is a big difference in PRRS virus ability to infect young pigs and old pigs. To remind you, virus grows rapidly to high levels in weaned pigs. Vaccine virus, it's attenuated, so it grows less well, but it still persists viremic for long periods of time. But you put the same virus into sows, you see it does not grow as well, and that viremia is curtailed much more quickly. And attenuated viruses are safer, they grow less well. These were not high-path PRRS attenuated viruses. So what is the mechanism that explains this inability of the virus to grow as well in older pigs? Well, if you look virologically, more virus grows inside the cells of young pigs. If you count RNA sequences in macrophages from young pigs, you see large numbers. Note that there is a lot of variation. Variation is something we will return to later. If you look at how many cells in a culture dish get infected, you see that the macrophages in young pigs are more susceptible to infection than macrophages from older pigs. And if you ask how much virus comes out of these macrophages, it's higher in adult pig and in young pigs than in old pigs. So by any measure, macrophages from adult pigs are more resistant to infection, and once they're infected, the virus does not grow as well. Okay. This difference has nothing to do with the ability of the virus to interact with the macrophage, because the receptor for infection, CD163, its levels are the same in young and adult macrophages. So we next asked if there were patterns of gene expression that were present in adult macrophages that were associated with ability to inhibit PRRS virus growth. Sean Wan did high throughput sequencing of the RNA in young macrophages and old macrophages. So young are green and old are red. And whether they were uninfected in the circles or infected in the triangles. And you can see that there is a big difference in the genes that are expressed in adult versus young, and a clear difference in uninfected versus infected. But of course, we want to know what these genes are, not just was there a difference in the pattern. So Sean did further analysis, and here we're looking at a comparison of the genes that are expressed in adult infected versus uninfected cells. So infected cells, we see 193 genes that are present in higher levels. And the one present in the highest level of all is interferon beta, major antiviral cytokine. And it's present about 120-fold higher in infected adult cells. That's the most differentially expressed gene. The next 9 are they're either interferons themselves or genes that are turned on by interferon. So adult macrophages have a strong antiviral response driven by interferon. What about young macrophages? Okay. Comparing gene expression in young infected versus young uninfected macrophages, it turns out we see the same pattern. Interferon beta is most highly expressed. The top 10 genes are interferons, or genes turned on by interferon, but there's a significant quantitative difference. The amount of interferon present in young cells is about 1/10 of the amount in adult cells. So they have the same pathway of response, but they appear not to be able to induce it as effectively. And this pathway is shown here. Pathway analysis is important and powerful in analyzing gene expression patterns, because cells do things by turning on series of genes. When a cell is infected with PRRS virus, it interacts on the cell surface, and there are signals that are picked up right there. But if that's the only thing that happens, there will be no consequence. Those genes send out signals that go through this entire pathway, resulting in effector functions. And here we're looking at interferon beta, which was turned on at a high level. But what's important here is you see genes all throughout the pathway being turned on. This is strong evidence that this interferon response really is important in antiviral immunity of pigs against PRRS virus. Okay. We think that this explains observations previously reported about the ability to isolate virulent PRRS viruses taken from the field and grow those in the laboratory. It's hard to understand PRRS virus if you cannot get the virus itself. And it turns out that if you try to isolate PRRS virus on MARC145 cells or MA104 cells, it's very difficult. Here we're looking at testing for virus on growth after 14 days. Only 2% of 50 isolates, that was 1 out of 50, grew successfully. If you put the virus on naturally permissive porcine macrophages from young pigs and look by fluorescence or cytopathic effect or RNA in the cells, you get higher success rates in isolation, but even under the best of circumstances, 30% of the isolates never grew. This is strong evidence that when those viruses were put on the cells, there was a strong antiviral innate immune response that prevented infection. Okay. So we can draw certain conclusions from studying the innate immune response in naturally permissive macrophages rather than in artificial model systems where you clone a protein and you express it in a non-naturally permissive cell line. We know now that there is a strong innate immune response driven by interferon. This sets the pig up to induce a good PRRS virus-specific immune response. It also has an important implication knowing that older pigs have a higher level of resistance. If you're using vaccination in a control program, it suggests that it's important to use full doses on a regular schedule, because if you take an attenuated virus and cut the dose, you may not succeed in getting an effective immune response. Okay. So now let's look at that adaptive immune response, which is critical for curing viral infections, both the initial infection and setting up a state of memory, which is how vaccines work. You know, if you see a virus one time, it induces a state of protection we call memory, so that when the virus is seen later on, you will get a robust response which protects you from that second infection. PRRS virus is different from other viral infections in pigs. Influenza, foot and mouth, Oyesky's disease are examples of many viral infections in pigs where you have a big increase in viral growth. The immune system kicks in and leads to a very successful eradication of the virus, such that there is no virus left in the pig after 2 or 3 weeks. PRRS virus, we know, is very different. You have a rapid increase in viremia. The decrease is not rapid. It takes weeks to eliminate viral infection detected in the blood. But even after you cannot detect it in blood, you have this long period of viral maintenance in lymphoid tissues, which can enable transmission and continued infection in herds. A second point here is this issue about variation that's important to me now in trying to understand the inability to gain complete control over PRRS virus. The viruses have, all PRRS viruses have certain characteristics in common, but they have other characteristics that are specific. One of those is how they grow in pigs. Here we're looking at average viremia levels in groups of 10 pigs produced by different viruses. And we see that while we have this standard model, there is a lot of variation in how individual viruses interact with pigs, including some viruses that grow very poorly. Duration of viremia can also vary a lot. It's interesting that these highly pathogenic viruses in the US and Asia tend to grow to higher levels and persist for longer periods of time in the blood, which is linked to their virulence. Okay. Even though we have a lot of variation and pigs have a difficult time controlling PRRS virus, we know that fundamentally immunity is completely effective. And that's been proven by herd closure practices, which are now used throughout the world, where if you have an outbreak and you fill up the barn with pigs, expose all of them to an attenuated or virulent virus, and wait for a long period of time, you introduce negative animals into the herd and they do not become infected. Functionally, there is no more PRRS virus in the barn, in the environment, or in animals. So if you give pigs enough time, the immune response is completely effective. This is a real important point for making vaccines because it says that the immunological principles work. And our task is not to figure out how to induce immunity that's effective when the pig cannot do it naturally. It's more along the lines of how can you make it work faster. Okay. This protection is cross-protective. I'm going to show you several slides here that emphasize this point, that if you give pigs one PRRS virus, they are protected against challenge with a different PRRS virus. And the examples we will look at are taken from exposing pigs to attenuated viruses, i.e. vaccines. So here we have an attenuated virus, and the challenge viruses are genetically substantially different. Percent similarity shown here. If you give virulent virus to these pigs, they have substantial lung lesions. If they have been vaccinated previously, we see a substantial reduction in lung lesions. So a lot of protection. This kind of effect has been observed numerous times in the laboratories of Boehringer Ingelheim. Here, Mike Roof presented data in about 2007 from emerging and re-emerging pig diseases meeting in Poland, showing that 16 different studies where pigs were given an attenuated virus vaccine, challenged with a variety of virulent PRRS viruses, lung lesions in the unvaccinated animals varied but were substantial. If they were vaccinated previously, there was a large decrease, sometimes to zero, in lung lesions in every case. So this observation of cross-protection is highly reproducible and variable. We have that variation thing again that we have to deal with. And Shishan Yuan has published this report showing cross-protection of a attenuated vaccine virus protecting against challenged with highly pathogenic PRRS viruses. Non-vaccinated challenged animals, 30% of animals died within 12 days. I'm always amazed by how pathogenic high-path PRRS really is. But 100% survival if they had previous immunity. In the same way, the clinical signs that were high, severe, and long-lasting were substantially reduced by previous exposure. So again, cross-protection, it's real. It's not 100%, but it is undeniable that it is present. One of the interesting things about this protection is that it does not conform or behave like one would expect. If you read textbooks in immunology, it says that this protective response, this memory that protects against future infection, can be demonstrated in the laboratory by seeing a large increase in antibody upon second challenge. This is called the anamnestic response. Okay? But in PRRS virus, if you take a group of 10 pigs and infect them with PRRS virus, you see antibody responses to all of the viral proteins. Here are the ones that we tested in this experiment, and you see there's variation from protein to protein, but there's a general response to all of the viral proteins. If you challenge the animal, nothing happens. There is no anamnestic response. And this is a highly reproducible observation. If you go into the field in a herd that is being vaccinated on a regular basis and you look at their antibody levels at a particular time, you see whether you're measuring nucleocapsid non-structural proteins, envelope glycoprotein on the right, you can see these have antibody levels that vary from pig to pig. If you revaccinate and look 35 days later, 247 animals, you see some titers go up, but you see a lot more not change or go down. On average, there's an overall decline in antibody levels. There certainly is no anamnestic response. Oops. And again, this is a robust observation. Here we're looking at each line. Oops, sorry. Each line is a group of 10 pigs. Okay. Infected with a different virus. So now we're looking at groups of pigs given a wide range of viruses that grow in different ways. If you re-challenge with a heterologous with a different virus, you see no change in nucleocapsid levels, or GP5 and 6, or NSP1, or NSP2, or NSP4. This absence of an anamnestic response is a robust observation, and it cannot be used to predict vaccine efficacy or the properties of any other viral tool for providing immunity. In fact, if you do see a large increase, what is the implication? The implication is whatever you did previously did not induce an immune response in the pigs. Even though we cannot see anamnestic responses, which are supposed to be what happens when you trigger memory, there is a memory response. That is shown in this study. Again, the first two, the green and the purple colored bars here, are what happens when you do in vivo, where you give one virus, or you give one virus and come back 2 months later with another virus. You see no change in the memory cells in different tissues of the pig: spleen, tonsil, inguinal lymph node, sternal lymph node, bone marrow. But if you take those cells out from these pigs and re-stimulate in vitro, where you just have a simple system of the memory B cells and the antigen, you see strong memory responses. So these responses are present in the pig for reasons we can speculate about. You do not see it when you give virus to the living pig. Okay. And if you look for memory B cells that make antibodies in pigs at times up to 200 days after infection, they're easy to find. You can find them against different viral proteins. You can find them in different lymphoid tissues of the pig, and you find them for as long as you look. Okay, so we know that PRRS virus induces a strong memory state in pigs. Skip that. I'm skipping the previous slide because it tells us that if you look in young pigs, you tend not to see neutralizing antibodies. There are dozens of published papers that you can draw that conclusion from. But if you look in adult pigs, which we've done now multiple times, you can find high levels of neutralizing antibodies. Here we're looking at 3 different herds, from 10 to 20 pigs, sows in each herd, and the neutralizing titers, if you dilute the serum out, you see you go from high levels of neutralization. It declines as you dilute. So the 50% neutralization titers are between 1 to 16 to about 1 in 300. These are substantial levels of neutralization. They are also cross-neutralizing because these pigs saw virulent viruses in the herd. They're being tested with VR2332, which is about 15% different. So this was the first good evidence that's been produced that antibodies have cross-neutralizing activities, and it's not that surprising since we have seen that PRRS protection is cross-protective in animals. Okay. Serum from these herds with cross-neutralizing antibodies really is broadly neutralizing. Here we're looking at the average result from 5 different sows. Ability to neutralize a range of type 2 PRRS viruses, and these herds saw type 2 PRRS viruses. Surprisingly, we see good neutralizing activity to type 1 PRRS virus as well. So again, this is truly broad neutralization. Okay. And these neutralizing antibodies are effective in controlling PRRS virus infections. If you purify these antibodies, put them into pigs and challenge with type 1 PRRS virus, shown here, you see a 41% reduction in viremia. If you challenge pigs with a type 2 virulent virus challenge, you see an 83% reduction in viremia. So these broadly neutralizing antibodies do have efficacy. They are able to help control viral infection. Okay. These antibodies are not directed towards the major envelope glycoproteins, GP5 and membrane protein, and this is not surprising anymore because it's clear now that these proteins are not required for the infection process. Okay. So we can draw certain conclusions now about innate immunity. It's clear that about adaptive immunity, the specific anti-PRRS virus response. That it's real, it's cross-protective, and we also know that while we have broadly neutralizing antibodies and we assume they have a role in protection, they are not required to gain protection, and that's the evidence that comes from growing pigs. Okay. Now, we're not quite done yet. Do you remember this slide? Again, the point here is that it's easy to draw lines and say this is what a response looks like, but that's just an average. In PRRS virus, we have to understand that there is a lot of variation. And this slide shows that if you look at the kinetics of individual viral infections, you see a lot of different patterns. Well, it turns out that the host pig also has lots of different patterns in how it responds. This is one example that demonstrates how differently pigs are able to respond to viral infection. Here, those different viruses in the previous slide given to groups of 10 pigs, those pigs had lung lesions shown here, where regardless of which group, where there were averages, some had mild lung lesions on average, some had a lot of lung lesions on average. Within the groups, you saw animals, one animal died, another animal in the same group had no lung lesions at all. You pick any group you want. You see some pigs that were hardly affected. You see other pigs that were severely affected. So in trying to figure out how we manage immunological control of PRRS, this variation is an issue we have to deal with. Okay. So here is a list of take-home messages, kind of literally taken from the preceding slides. But the overall message that's important to understand, first of all, is that in looking at the host-pathogen interaction, there's enormous variation in the viruses and in the pigs. And what we knew 10 years ago, some of that does not apply. The viruses we have today largely did not exist 10 years ago. 10 years ago, you just started seeing high-path PRRS. Now you've got other viruses. 10 years ago, the genetics of pigs were different than the genetics of pigs today. So you have this constantly moving target. But even so, we know that immunity works. We know that it is cross-protective. We know that there are differences in animals that affect how well exposure to PRRS virus induces a memory immune response, but we know that memory is there. So the consequences of this memory is that an immune pig, immunized with vaccines, for example, you will have a reduction in the amount of disease that's caused. You'll have a reduction in the viral load in the pigs, which leads to a reduction in the amount of virus shedding, which reduces the amount of transmission. So by all measures, immunity improves the herd status of pigs, but we also know because of variation that if you're trying to use attenuated viruses which are safe to improve the health status of pigs in the presence of PRRS, you need to use full dose products on a regular schedule to provide the most robust coverage. If you cut virus doses, if you take shortcuts for reasons that are not related to accounting for variation in your herd, variation in your viruses, and the changing dynamics of herds that occurs all the time, then it's more difficult to achieve immune protection, which is one component of overall control of PRRS virus. Thank you.